So today we will discuss about osmoregulation in water, right? Our topic is osmoregulation, right? So now basically there are two types of organism. The first are known as osmoconformers, right? And second are known as osmoregulators. Osmoconformers and osmoregulators, right? So basically osmoconformers are the organism whose body fluid is isotonic to the external environment. When body fluid, basically these are the organisms whose body fluid is isotonic to external environment. Basically these are the organisms whose body fluid is isotonic to their external environment. Whatever their external environment is, their body fluid will become like that, right? But osmoregulators are organisms which have different body fluid to external as compared to external environment, right? So basically osmoconformers are the organisms whose body fluid is isotonic to their external environment, and osmoregulators are the organisms which have different body fluid as compared to the external environment. Their environment can be hypertonic, their environment can be hypertonic, their internal, or their environment can be hypotonic, but it will be opposite to the external environment, right? So basically we have two organisms: the organisms who live in fresh water and the organisms who live in marine water, right? So today we will discuss about the organisms who live in fresh water. Basically osmoregulators in fresh water. Osmoregulation in fresh water, right? Then basically consider that we have amoeba. We have amoeba. So in fresh water, basically the external environment is hypotonic. The external environment is hypotonic and the internal environment is hypertonic. Basically the internal environment has high osmotic potential, high osmotic pressure. The internal environment has high osmotic pressure, which means that there is high solute but it has low solvent. So we know that water always moves from high solute concentration to low solute concentration. We know that water moves from low solute to high solute, right? So it means this amoeba will feel flooding. It will feel flooding. So the water will come inside this, right? In order to manage this, basically in this amoeba there is a contractile vacuole. There is contractile vacuole. There is a contractile vacuole which basically helps to remove the excess of water. This helps to remove excess of water, which basically helps to remove the excess of water, this contractile vacuole, right? So I hope this osmoregulation in amoeba is clear to you. Then next organism which we have is basically fish. It's basically fish. Okay. Consider this is fish, and as I said that this fish is in hypotonic environment. So the external environment is hypotonic and internally it is hypertonic, right? It means that water will come inside this fish. Water will come inside this fish and the fish will feel flooding. So what will happen? Dilute urine will be passed. Dilute urine will be passed, but in that dilute urine, excess of salts will, basically essential salts will also be lost. Essential salts will be lost. So in order to recover those lost salts, active transport will take place. And in this active transport, some special cells do this active transport of salts. Those special cells are known as ionocytes. Ionocytes are basically the cell which do active transport, which actively transport essential salts, so that the body can recover the essential salts and the water in a solute level is balanced, right? So I hope this osmoregulation in freshwater fish and freshwater amoeba is clear to you. If you have any kind of queries, you can ask me in the comment section. I'll be really glad to answer you guys.